lineup of speakers. Ellie and I tried to pick themes that span the breadth of what AI could be contributing to cancer in general. Obviously, we're focusing on kidney cancer, but we're bringing investigators that have thought about this from different perspectives as well. So the first one I'd like to introduce is Sylvan Baca from Dana-Farber. Yeah, and I guess the speaker should stay here at the end of their talks. Oh, yeah, I guess so.
Okay, does this work? Good.
Yes.
All right, well, thanks a lot to the organizers for putting together this conference. And all right, let's see if this works. And thanks for the invitation to speak here on this session, which I think is really timely and interesting. So, all right. So I was asked to give a brief overview on AI and cancer genomics, and it's obviously a very big topic, but we'll see how far we get in 7 minutes. These are my disclosures. And as you might have heard, there's been a big explosion in AI research in oncology in the last several years. This plot here is meant to just convey kind of the scale of it. If you look at yearly publication counts of original research articles on AI and oncology, they've trended upward consistently since 2018, and this is probably a major underestimate, but just to give a sense, this is a very active area of research. And I think one reason why oncology has been an appealing place to apply advances in deep learning methods and other artificial intelligence strategies is that it meets some basic requirements of a good AI problem. So oncology has large data sets, which is something that AI applications usually need to shine. When we treat patients, we generate lots of data in terms of things that go into the medical record, genomic data sets, imaging, pathology, and all this can be used by AI models to predict labels that we care about, which could be things like, will this patient respond to treatment? Does this patient have an EGFR mutation? And we have features that associate with these labels in meaningful ways that allow us to make accurate predictions. So I'll give some examples of how AI is being incorporated into cancer genomics, and this is certainly not an exhaustive list, but just a few examples to highlight the point. AI has been used to enable better calling of mutations in DNA sequence, better prediction of pathogenicity of variants when we see them in DNA, and it's also been used more recently to infer the presence of mutations and transcriptional features of cancer from histopathology slides, which I think is a very interesting application. And it's being used as well for clinical subtyping based on multi-omic phenotypes. So to start with improvements in variant calling, on the left is some work from Dr. Van Allen's lab from back in 2020 that showed you can use deep learning methods to look at sequencing reads from cancer and do a better job of identifying true mutations and distinguishing them from artifactual variants. And applying this approach to a germline sequencing data set found quite a few additional potentially pathogenic germline mutations that could predispose to cancer risk compared to the traditional methods that we'd used prior to this. And thinking about the liquid biopsy circulating tumor DNA space, which is where my lab does a lot of our work, this has really been enabling by applying deep learning methods. This is an example from Dan Landau's work to do a better job of distinguishing true variants from artifacts. And this is important in circulating tumor DNA, where we're often looking for a very small number of mutated tumor-derived molecules in a sea of non-mutated, healthy-derived background DNA. And the improvements that have come from using deep learning in this setting are real, and they've really enabled high sensitivity, minimal residual disease applications that surpass what we could do just several years ago. We can also do a better job of understanding which variants that we detect in cancer are likely pathogenic. So in the early days of cancer genome sequencing, we had many of these variants of unknown significance where they introduce a change into protein coding sequence, but it's hard to tell from the context whether that change actually has a deleterious effect on the protein function or changes its function in some way. So this is a tool that was developed using AI by a team at Google who's pioneered a lot in this space called Alpha Missense. And what it does is it looks at a number of different features, including some protein language modeling, some structural predictions from AlphaFold, which is a protein structure prediction AI method that Google also developed, and also population frequencies variants. And it puts all this together in a machine learning model to predict, is this variant pathogenic or not? And when it's applied to unseen variants in ClinVar, which is a database of human genetic variation and its functional impacts, it does a good job of distinguishing pathogenic from benign variants. Now, I think an interesting place where there's been a lot of effort recently is seeing if we can predict the presence of mutations from histopathology imaging. And the motivation here is that if you could do this, potentially, for example, in resource-limited settings, you could forego the need for DNA sequencing. And the premise is that these changes in DNA sequence may cause subtle changes in the appearance of cancer cells or in the tumor microenvironment that we can see in slides and potentially infer whether a mutation is present without DNA sequencing. And one of the first papers to demonstrate this concept was back in 2018, where they looked at lung cancer and tried to predict the presence of several driver mutations. And a lot of work has been done since then on other cancer genes, other cancer types, and some have claimed higher accuracy than is shown here, although there are some caveats to these approaches that we could get into. But I think one interesting way that this is being applied in kidney cancer is in detecting a genomically defined subtype of kidney cancer. That's translocation RCC. This is a rare aggressive subtype that is driven by a fusion of one of two transcription factor genes. Histologically, it can look a lot like clear cell carcinoma. So this is a place where using AI to pick up subtle differences in the appearance on slides can be useful. So this study basically trained a predictor to look at H&E images and guess whether there was a translocation present that defines this rare subtype, and it did a pretty good job of identifying these cancers. And taking this a bit further, some groups have been interested in inferring gene expression from H&E images, and this is very much an active area, but just to show one example, this was a paper that looked at these whole slide images and tried to predict expression patterns using TCGA data where we have matched RNA-seq and images from the same cancers. And you can see that their predictions are only modestly correlated with the ground truth measured from RNA-seq on unseen samples, but they do seem to capture some expression patterns in biologically relevant modules and contexts like angiogenesis or hypoxia in kidney cancer. And taking this a step further, this was a study that used this concept and tried to predict, based on the appearances of cancer under the microscope, the value of this angiogenesis score that's based on RNA-seq that had been previously reported. So they trained a classifier to predict the angiogenesis score just using slide images, and they showed pretty good correlation with the observed RNA-seq score and what they could predict based on how the tumor appears under the microscope. And these scores actually were very prognostic in that patients with a high angiogenesis score tended to do a lot better in terms of overall survival and other outcomes compared to those with lower scores. So there are some caveats here too that we should keep in mind, particularly as this field develops, given that it's relatively new and a lot of these methods have not been validated prospectively. I think one important one is that these are very high dimensional data sets, and a lot of mutations in cancer are highly correlated with each other. And the same is true for expression patterns. And so it's got high potential for confounding, and it's hard to know when we're running these algorithms, are we really predicting the isolated presence of a particular mutation, or are we picking up on something that's correlated to that mutation? That's a hard problem to solve, but people are actively working on it. So to wrap up, I'll just offer a few thoughts on future directions. I think prospective validation is very important in this field. I think it's comparatively easy to train these models on existing data, but to really show that they work on data that have not been seen before and haven't even been generated, or generated on different platforms in different contexts, is a very important step for actually allowing researchers and clinicians to trust and use these algorithms. I think we need more diverse representation in the training and validation data that we use. A lot of these tools have been trained on consortium data like from TCGA that tend to oversample white and European ancestry patients and don't really reflect the diversity of the US population, let alone the world. I think it'll also help a lot to have interpretability in these models. I think many of the early models have sort of been critiqued as black box, and there's a trend toward trying to have models that can explain to you how they're making their decision, and I think that'll help gain trust and uptake. And I think it's important also to really accurately capture uncertainty in the predictions, which has been challenging. And finally, most of these models are prognostic, and I think it will help to move toward predictive models that can help us decide not just how is this patient going to do overall, but what patient is this, or what treatment is this patient most likely to respond to. And with that, I'll wrap up, and thank you for your attention. I'm looking forward to the discussion at the end.
Thank you, Sylvan. That was fantastic. Our next speaker is Dr. Julie Stein-Deutsch from Johns Hopkins about AI and pathology.
pathologist at Johns Hopkins University, and I'm very excited to be part of this session. I'm going to discuss a little bit about incorporating AI into pathology. So this is a little bit of an overview of how AI is currently being thought to be used in pathology, largely falling into three buckets. The first is quantification, which I'm going to spend most of my time talking about, which is automating tasks that pathologists do every day. That includes things like PD-L1 expression, assessment. The second is prediction, which Dr. Baca just gave a nice overview of how we are thinking about using routine H&E or routine histology slides to predict molecular signatures. And then the third is sort of agentic, more generative AI type processes. So I really like to think about AI as facilitating computational pathology at scale. And for this brief talk, I'm going to focus mostly on a use case, which is where I think computational pathology is really shining, and that's in pathologic response assessment after neoadjuvant therapy. So now as we move towards treating earlier stage cancers and giving therapy before surgery, we have this need to assess how well the tumor responded after therapy. And the way we do this is by assessing the percent residual viable tumor, and that's a surface area measure of the estimate of residual viable tumor over the tumor bed area, which is comprised of three classes: regression, residual viable tumor, and necrosis. And really, what is regression? So, the two other classes, tumor and necrosis, are pretty explanatory, and those have been relatively well-defined concepts in the field. What is tumor regression, or how do we define where tumor used to be that's now been cleared after therapy? And we have spent a lot of time studying pathologic response, from the initial neoadjuvant anti-PD-1 based studies in lung cancer and melanoma. In 2018, we were part of the first in-human anti-PD-1 neoadjuvant trial in lung cancer, led by Patrick Ford at Hopkins. And as part of that study, we identified this constellation of features that really characterize tumor regression. And those features include immune activation, so tumor-infiltrating lymphocytes, tertiary lymphoid structures, plasma cells, as well as features of tumor cell death, so cholesterol clefts and foamy macrophages, as well as features of tissue repair or even scar formation, so new vessel formation and fibrosis. And after looking at features in lung cancer and melanoma and seeing that they were virtually identical under the microscope, we then looked across tumor types. And lo and behold, the regression in these various tumor types looks virtually identical even to the non-pathologist's eye across these different tumor types. So you can see renal cell carcinoma there on the end. So these were tumors treated with neoadjuvant nivolumab. Similarly, we can look at on-treatment biopsies in patients with advanced metastatic disease receiving immunotherapy-based regimens, and we see the same exact features of response. Here again, highlighting a case of renal cell carcinoma. And so in 2020, we proposed the initial pan-tumor pathologic response criteria system, which is effectively the pathology equivalent of RECIST. So RECIST has worked quite well in the advanced metastatic disease, and unfortunately it has a lot of limitations in the early stage neoadjuvant setting, where pathologic response has now emerged as the gold standard metric of response. In 2020, we published this initial pan-tumor proposal, which we have now updated and published earlier this year in 2026. And part of that had to do with the lessons that we learned from the PD-L1 testing landscape. So this is what a pathologist has to contend with when they are scoring for PD-L1. So this is a table which represents all of the different tumor types and assays and scoring criteria and cutoffs that vary. And this has been a huge headache for pathologists and also for clinical trialists trying to compare across studies. And so we've really operated under this guiding principle that tumor type specific scoring systems of response would have to clearly be superior in predicting outcomes for patients to really warrant having these disparate scoring systems across tumor types. And pathologists are really reproducible at scoring across tumor types using the pan-tumor scoring system. We partnered with SITC for a international multi-center study looking at 14 pathologists scoring across tumor types, and they were very reproducible in a scoring pathologic response. So what's the problem? We know that pathologic response is great and it has shown great promise in predicting outcomes, but it doesn't scale easily. So even on the glass-based or even using digital annotation tools, this is what it looks like to annotate a slide on glass, where you have to circle the different regions of the tumor and the regression bed. This is time-consuming, it's subject to pathologist variability, and it requires an expertise that's really outside of routine pathology training and is mostly centered at academic centers where a lot of those initial trials have been done. So we built in 2022 a machine learning-based model that recapitulates a pathologist's ability to perform pathologic response assessment. Here you can see across the spectrum of residual viable tumor, our machine learning algorithm overlay of predicted regions for each tissue class. So in red is the tumor, blue aqua is necrosis, and green is the regression bed. The solid lines are the ground truth annotations by the pathologist, and you can see the shaded regions which show the machine's prediction of each tissue class. And you can see the very strong correlation between the pathologist-assessed residual viable tumor and the pathologist, the machine-assessed residual viable tumor, and this was our initial pilot study that we did in neoadjuvant treated lung cancer. So there are a lot of things to consider when thinking about developing a model and optimizing a model. That includes how do we best pre-process slides and whole slide images, how do we optimize the tile size, which accounts for different magnifications that pathologists use in their daily practice, whether to use overlap regions and by how much, and how to weight, with probabilities, different features or tissue classes. So future directions, we are extending our machine learning algorithm to 10 different tumor types, including kidney cancer, and so I hope to be able to share those findings with you at a future meeting. And we are really, in my group, focused on utilizing AI to really leverage H&E-based biomarkers. H&Es are standard routine slides that are generated all over the world, and they can be used in under-resourced settings especially, and so they represent a very remarkable source of information. So with that, I'd like to acknowledge my collaborators at Hopkins and elsewhere and funders. Thank you so much.
Thank you, Dr. Dush. Please have a seat. Our next speaker is Dr. Smith, a radiologist who joins us from St. Jude's Children's Research Hospital, also a prior KCRP recipient, and he'll be talking about the role of radiomics and AI in kidney cancer.
Yeah, thanks. Delighted to be here. Appreciate that. So we're going to talk about AI and radiology. We're early adopters of this technology for our field back in the machine learning, deep learning area, and now we're in this generative AI area. We'll sort of cover some of that. So these are my disclosures. Let's first talk about imaging biomarkers. You know that a biomarker in imaging is a quantitative imaging measurement that reflects biology or predicts meaningful clinical outcomes. And things that we want to see, we want something that's really simple, objectively measured and quantifiable, associated with something that's meaningful like a clinical state or outcome, and reproducible across observers, scanners, and institutions. I find it tends to be very helpful if these are handcrafted, which is really derived from biologic or clinical knowledge. And a great example of this is tumor size. Think about how size is related to outcomes. So let's talk about that. It's a handcrafted quantitative measure of the amount of tissue. This is from observed associations. It's really simple math. 0 actually means 0. You can calculate percent changes. You can add things up. It's widely applicable to almost any tumor. It crosses imaging modalities, even CT versus MRI, with or without contrast. It crosses institutions. It's strongly associated with outcomes. If you look at this tumor here, this is anti-angiogenic therapy, pre and post therapy, left versus right, and you could measure the size, and maybe it underestimates what we're actually seeing with this, but look at the red color, which is is another size metric. You could look at the area of vascularized tumor, the volume of vascularized tumor. Those are all size metrics. Classic linear size measurements tend to have some weaknesses around internal devascularization, and we've seen with pseudo-progression and immunotherapy that's another area where it potentially fails. Let's look at that compared to radiomics. This is a discovery-driven approach that extracts hundreds to thousands of metrics of quantitative imaging features to identify candidate imaging biomarkers. So see the difference here? We're looking at the images and then trying to associate with biology after the fact. And so there's some strengths to this. There's high throughput biomarker discovery. It supposedly can capture hidden features in the images, and it integrates with machine learning and multimodal data. And these are examples like a gray level occurrence matrix, a gray level occurrence, and there's probably thousands of these type of markers you can do, and then you can apply image filtration and get to tens of thousands from a single circle around a tumor. That actually comes with major problems. This high dimensionality increases the risk of a spurious association. So looking at 100 tumors and tens of thousands of biomarkers, and of course it's going to associate with something, but is it going to be reproducible? So these features often lack explainability and biologic interpretability. They're sensitive to acquisition, to reconstruction, to segmentation. They are essentially not reproducible from one center to the other. And I would argue that none, and maybe at best few, have ever achieved external validation and clinical translation. So I may have hinted at that I'm not a super fan of radiomics. But compare that to artificial intelligence. These are algorithms that learn patterns from data to perform tasks that typically require human intelligence. Some clinical applications that I will show you momentarily are image reconstruction, segmentation, tumor response evaluation, opportunistic screening, and then we'll talk about clinical decision support. Now, what do you need for these things? What's required clinically to be successful for AI? You need this multi-institutional external validation. You need regulatory clearance. It needs to be integrated into the workflow, and it has to demonstrate clinical value. So here's an example of one. So the image on the left, which is nearly identical to the image on the right, the one on the left is conventional, no AI, and then we're using AI to speed this up. And the colors represented various sequences that might be in a renal MRI exam, for example. And so you can speed that up. You can also improve the quality. If you look at like the butterfly wings, I'm trying to show you improved resolution. You can also do that with MRI. I find that these algorithms that I've seen commercially tend to do better at speeding the exam up and not so much on actually improving the resolution in a meaningful clinical way. But you can get to 20 to 80% faster. It depends on the type of exam and what you're doing. This leads to fewer motion artifacts. I tend to see more higher quality that's more signal to noise related than I do to spatial resolution, and you can get higher throughput. Who here has ever had trouble getting their patients on an MRI scanner? This opens up that opportunity if we can move patients through faster, because it's not just in renal cell, it's across the board in MRI. Tumor segmentation is another area. You can, of course, segment the tumor in yellow here, but look how we've segmented everything around that. This is now possible for artificial intelligence. This is a Wilms tumor, bilateral, that's in yellow there, but notice we've done the kidneys, the collecting systems, arteries, veins, etc. This is really useful. We can do automated measurements that I'll show you as an example in the next slide. You can also do surgical planning. Our surgeons put virtual reality goggles on, do the planning prior to surgery, and they really like this. They almost feel they can't do the surgery without it at this point. And I think it enables scalable quantitative imaging. We've heard that word before this morning about scalability with this. Here's tumor response. We saw a little bit of this at the introduction by Brian. On the right is the prior exam. On the left is the current exam. The human is interfacing with this AI, and there's two different types of AI. One that's going to help us track the tumor, and then one that they're going to click or draw a box around the tumor to activate AI to measure it. It almost happens so fast it's hard to tell what's going on, but it helped track that lesion down. The user just clicked on it and measured it. It's going to track the next one. When they hit a next button, it just jumps from tumor to tumor to tumor. You can't forget anything. It semi-automates the process, probably improves inter-observer agreement. So here's a liver tumor, and we just click on it. You can edit it. It auto-zooms in, these kinds of things. Some of the advantages of this is that it's very standardized. It leads to fewer errors. The most common error in radiology is data transfer error, where I say 3.5 centimeter, it writes 1.5 in the report, and nobody detects that. There's no missed lesions. You cannot forget a lesion. It won't let you skip a lesion that was previously identified. It's faster to do this. It doesn't require any dictation, and it ultimately leads to improved communication. So what do I mean by that? You know, you're used to a normal text report coming from your radiologist. How about a graphical report? How about percent changes automatically calculated, a table, key images, everything that you need to be able to communicate with your patient? And you can imagine in the future converting this into any language or any health literacy. AI opportunistic screening, I'm not sure how familiar your field is with this, very common in ours. This is asking the question, are there other biomarkers besides the tumor on these routine imaging exams, particularly CT as an example? So yes, there are. There's tons of things that we can fully automate in the cardiovascular space and metabolic space. We heard even from our speaker this morning about the importance of obesity. Imagine if you could get down to levels of visceral subcutaneous fat, bone density, muscle, etc. And that's exactly what you can do. You can do this on every single CT scan at your institution in a fully automated way. Data is available in like 5 minutes off the scan. You can look at coronary artery calcifications, the size of the heart, which is a measure of cardiomyopathy, visceral adiposity, sarcopenia, bone density, etc. can be instantly gathered using these kinds of techniques. And this is really the concept that AI opportunistic screening, I think, is going to impact treatment care as we, both at the beginning and throughout the course of therapy. The future of kidney cancer is probably multimodal. What do we mean by that? We mean the different data sets that we just heard our other speakers talking about. That's the radiologic imaging, the pathology, genomics, laboratory values. All of the clinical data in the EMR is accessible. It's all digitized. You can bring it all together with multimodal AI. You could add a layer of clinical decision support on that. Theoretically, what it could do? It could help us choose optimal therapy, think about predicting response, identifying toxicities, and prognostic information on survival. And I think this kind of material will be available in probably 2 to 5 years. That we'll start to be able to see these things translating at certain centers. So this is data-driven, patient-centered, personalized kidney cancer care. I think it's coming, especially for groups that are amassing their data sets together. So we put all of our data sets into one high-performance computing network so that we can do these kinds of things. So we talked about imaging biomarkers, wanting them handcrafted, quantitated, super simple. We said radiomics, that doesn't quite meet that. Currently kind of failing after 10,000 plus studies, not seeing it translate very well. But AI is really making it big. We saw it in MRI, we can do it in CT, PET CT. We use it for segmentation of tumors. We use it for tumor response evaluation. You can use it for opportunistic screening. And I think the future is going to come with this multimodal data and clinical decision support. Thank you for the opportunity, guys.
Last speaker for our rapid fire presentation for the session is Hari Kazi from Almara about clinical trial discovery.
Hello everybody, I'm Hari. I'm the CEO and co-founder of Almara. Almara and KidneyCAN have partnered together to bridge the gap between patients and clinical trials. Cancer research is a pivotal piece of accelerating cures, and that's our goal. So the KidneyCAN Trial Navigator platform. So let's look at a few snapshots of what a patient may experience while using this application. So first, a series of questions. These questions are catered to be both comprehensive, to collect medical information, as well as cater to patients of all backgrounds. After entering this information, you also provide a city and a search radius, and this allows you to find trials within your vicinity. You see both matched and ineligible trials within your radius, and you're able to see why this trial was found or why this trial would be ineligible for your case. You're able to see study drug data that's been publicly published, as well as more information about the trial, which includes things like the criteria and description, and these are also available in simplified formats for patients to view. The goal is for patients to be able to export these results, either print them out or email them and take it to their physician during their next visit. So let's talk about the technology that goes into something like this. So first is the data. So where do we source the data from? The data comes from both the NCI API as well as clinicaltrials.gov. We do this step called augmentation. We take the data and we extract all the key pieces that we need for matching, as well as identifying the target patient population for that trial. Then we enhance the data with things like site location using Google Maps APIs, study drug data that we're able to scrape through APIs like Perplexity, as well as simplified formats for patients to view. All this information gets fed into our matching engine. There's two ways to make a search on the platform. There's either the questionnaire that you previously saw, or there's a free text box option where you can put in any natural language query. This is then fed into our LLM to create a patient profile. And first, we retrieve all the trials within the patient's radius. Then we're able to actually do the screening where we use another LLM to take the patient profile and the trial's target population to try to identify if this patient's a fit. Then we're able to re-rank the trials based on the study drug's appropriateness, safety, efficacy, and all these other profiles. Then the patient is presented with these matches, and along with this, they're presented with a visual map where they can see the proximity of each of the match sites, as well as how many trials are outside of the radius to see if they're willing to expand their travel. I'm excited to share some data from a pilot that we were able to conduct with 31 patients. So patients and caregivers were survey respondents, and we found it interesting to learn that 53% of survey respondents actually had not heard of clinical trials as a treatment option from their physician. But they were still willing to give this platform a chance and give it a try. And out of the patients that answered this survey, 62% responded with active disease that they found a trial on the platform that that'd be worth considering. 74% of patients also found the layout, the usability, and all the information presented to them on the platform to be easy to use. KidneyCAN also plays a pivotal role in how patients perceive this platform, and 84% of patients and respondents said that KidneyCAN's role increased their trust. We also found it exciting that 45% of patients and respondents said that they were more confident in discussing trial options with their physician after using a platform like this, and that 94% of them would actually take their results to their oncologist, allowing them to start the conversation with their physician. So what does this mean across the clinical trial ecosystem? This means that patients are able to bring a shortlist to their visit. They're able to initiate trial conversations with their providers earlier in their treatment journey. This platform also allows for community and academic oncologists to have a tool to search for trials quickly. The natural language form of input that I described earlier is a perfect way to enter quick queries to identify trials at referring sites or at other locations. And for pharma, this means this is an opportunity to surface patients in community settings that might not have trials as an option, but presented in their typical care setting, as well as being able to show trials to patients at a key decision point for them. With that, I would like to thank all the oncologists that helped look at this platform and provide their feedback, as well as all the survey respondents, as well as KidneyCAN for this opportunity to present. If you have any questions, would like to see more data from the survey or a demo of the platform, we'll be sitting at the table right outside and happy to answer any questions. Thank you.
All right, we have about 7 to 10 minutes for questions if anyone would like to ask any of our speakers, so I'll stay up here.
Another one. Well, while people are getting warmed up, maybe I'll just start with one, quick one. You know, there's a lot of buzz these days around AI agents. Curious if any of you are using them in your research or clinical care, or maybe also for the audience to explain to folks what, if they don't know what that is, and where you see it's going. Where you see it going in this space? Any takers?
Dr. Deutsch?
So one, well, we got.
Yeah, go ahead.
I'll take one. So we're doing AI agents are things that automate various processes. So you can think of it, that's probably the most common use of them. And one of the examples I gave, like opportunistic screening, there are groups out there that are deriving this data using classic machine learning or deep learning off of the images, whether it's coronary artery calcifications, body composition, and these things. And then you can automate downstream care. You can route it to the right people. You can pull information out of the electronic medical records. So it's usually like a multimodal thing where it's pulling data together and directing it to the right clinic for the care.
Dr. Deutsch, I know some people are talking about cytology, that they're being kind of automatedly done by agents and then triggering the pathologist to look at those. Have you come across that at all in the pathology world?
Yeah, so I think there's a lot of potential use cases for obviously automating any sort of read, whether that's in cytology or elsewhere. I think it's only going to serve to really accelerate and facilitate pathologists' ability to review things quickly and accurately. I don't personally review cytology in my practice clinically, but certainly I can see that there's a lot of merit to that for sure.
Dr. Hammers?
Put the terms on. I don't know. How's the training going these days? Meaning, is it still the old-fashioned machine learning, and we have the thousands of different scans, and then slowly the system is getting better? Or are things changing with these LLM models who are becoming now almost omniscient, if you will, and are becoming, let's say, a really good pathologist, really good radiologist, or something of the nature? Like, what's the starting point these days of data sets that we need to have to get good at something or find something meaningful that we can work with? If you could help us with that.
Yeah, I mean, I can start with that on the pathology front. It depends on what your goal is, and I think it depends on where you're starting from in terms of your expertise. Certainly, we're getting better at using, fewer slides, for example, in training. I mean, for deep learning models where you're really putting into black box AI and trying to come up with some sort of signature, you need a lot of training data, a lot. For us, I mostly work on the supervised side of things where actually if you put in really good data to the model and really well annotated data, you don't need that much to actually train a really strong model. But again, when you don't know what you're looking for and you're just trying to put, plug and chug everything into AI, you do need a lot of images, at least on the pathology side.
It's a little bit similar in radiology. I would say, like, for body composition, you would hand annotate that. Maybe 1,000 cases would be enough. Get into tumors, like, depends what your goal is. If you want to guide the tumor to measure something, easily 1,000 would be enough. If you want it to be fully automated, closer to 10,000. So it just kind of depends what you're going after. Detection and automation require larger data sets, hand annotation. When you get into the LLMs, a lot of times you can deal with dirty data. Dirty data is okay. There are ways to mitigate that, and you're going to kill it by having so much higher volume. So bringing it together, finding ways for us to cross with each other and communicate and share data, that's going to be really critical to advance this field.
Do you want to ask Sylvan?
Oh, we have one last question. Yep. So I'm happy to just chime in. So I completely agree with you. I think it's part of a general challenge in the AI biomedical world around benchmarking, because if you look at a lot of, let's say, the pathology foundation model studies that have tried to aggregate a very large corpus of pathology images to train to make generative AI, they can, the benchmarking that they use are tasks that this audience would find pretty easy. How good is the model at distinguishing clear cell, papillary, and chromophobe RCC? I could probably sit down with my 9-year-old, and just start to show some pictures. At least she would start to get it, right? Which would not be perfect, but you know what I mean. Like, they're very obvious. Like, they look very different from each other, but very nuanced things are harder. And that benchmarking issue is pervasive, whether it's pathology, radiology, but it's also pervasive in the biomedical sort of preclinical space as well. All of the single cell foundation models that have been generated do not perform very well in niche cancer-specific use cases, at least so far. And in those domains, we are like billions or trillions of pieces of data away from actually having robust generalizability. So it's actually it's a big problem, generally speaking.
Talk very generally about it, but we can't forget that we're far away from these points where details matter, and they matter most, especially for patient care, on those which are the outliers, or the ones who are further from understanding.
So on that note, I think we are calling it. We're going to pass the baton back to agentic Tony Shueri, I think, who is about to know.
No, I think is Brian.
Just kidding.
We have a break now, Brian. Yeah, I think Brian's going to come in. All right.
But
Thank you for
and
all
the
our
panel.
speakers. Yeah.